to SARC Talk. SARC is a nonprofit cancer research organization that develops and manages clinical research trials in pediatric and adult sarcomas. SARC Talk is a podcast to discuss activities of SARC, interview sarcoma research leaders, and review scientific initiatives in the sarcoma field.
Welcome to SARC Talk, episode 25. My name is Scott Okuno, a medical oncologist at Mayo Clinic and chief medical officer at SARC. At SARC Talk, we interview interesting people who are connected to sarcoma, where we can all share and spread the knowledge about sarcoma. All episodes are posted on our website, Spotify, YouTube channel, or wherever podcasts are available. Please subscribe and put comments down because we do look at them. Today, our guest is Dr. Margaret von Mehren, Professor of Medical Oncology, the Department of Hematology Oncology at Fox Chase Cancer Center. She is the Physician Director of the Clinical Trials Office and Associate Director for Clinical Research at Fox Chase Cancer Center, and she is also the Director of the Sarcoma Program. Dr. von Mehren did her undergraduate at Georgetown University and her medical school at Albany Medical School. She did her internal medicine at New York University Medical Center, and then she came to Fox Chase to do her fellowship in medical oncology, where she's remained on staff. Please welcome Dr. Margaret von Mehren. Welcome, Meg.
Thank you, Scott. It's nice to be here.
No, Dr. von Mehren, can you share with our audience how you decided to get into oncology, and more specifically, how you decided to do sarcoma?
Yeah, so when I was in high school, I definitely knew I had an aptitude and interest in science, particularly biology. And so I was sort of looking and thinking about what I might do when I grew up. At the time, I found I had a really challenging time in chemistry in high school. And so the idea of having to pass organic chemistry sort of dissuaded me from thinking about going to medical school. So I actually started in nursing school, and I had this path set out that I was going to become a midwife. That was my goal. And then during chemistry in nursing school, because you have to take chemistry in nursing school, I actually figured out chemistry and ultimately decided to go to medical school. And when I was doing pre-med chemistry, if I didn't want to do chemistry, I got my fill in college. My professors started talking about using radioactive elements for treatment of cancer, and it really sparked my interest and curiosity, and that's where my interest in oncology started. And every time I came across something that had to do with oncology, it just kept intriguing me, and I entered medical school knowing I wanted to be an oncologist. I didn't know if I wanted to be a pediatric oncologist, a medical oncologist, a surgical oncologist. I just knew I wanted to be involved with cancer care. And as I went through my training, I spent a summer actually at Memorial working with one of their pediatric oncologists. His name was Brian Kushner, who was a specialist in neuroblastoma. And we would have almost this daily discussion about pediatric versus adult care that summer. And the thing that I struggled with, and probably as a future mother, was you not only have this child who Dr. Kushner would tell me, they're so resilient, they bounce back, but you also have these parents. And I found that dynamic very challenging. And so ultimately, I decided to be involved in adult oncology care. And although I loved surgery, I ended up liking patient rapport and interactions in a way that I thought surgery was not the best fit for me. And so went into medicine and ultimately medical oncology. So that's how I got to where I am today.
Yeah, so first of all, I forgot or I don't remember about the nursing aspect going through there. And if it was not for chemistry, you might have started out in that field, but you eventually got here. How did you end up in sarcoma? Because medical oncology is very broad.
Yeah. So when I was in fellowship, I actually was really interested and jazzed about immunotherapy. So I actually spent time in a research lab working on trying to develop or think about cancer vaccines, and worked on trying to develop a HER2 mouse model, and did start and was funded to run some vaccine trials targeting CEA. But I think I was probably sort of a decade ahead of where sort of major breakthroughs in immunology were happening. And so while I was intrigued, there just wasn't a lot of there in what I was able to accomplish. As a senior fellow, I was in sarcoma clinic, and my supervisor there was Dr. von Romling, who I had known as he briefly in medical school. He had been an adjunct faculty at Albany as well. He worked at Sanofi, and so this was his clinical outlet. And so when he ultimately moved away from the Philadelphia area, I was sort of handed the sarcoma program because there was nobody else. And so at the time, I had kept on, when I joined the faculty, I'd kept on seeing sarcoma patients. And I think one of the things that I noticed compared to my breast cancer clinic, which I also did for a while, there were a couple of things. Number one, sort of the patient population, there were men and women. There are male breast cancers, but that's very few. And each case was so unique and different. It could be anywhere on the body. Patients were young and they were old. So I really liked the patients. And then I loved the biology behind many of these sarcomas. I also found on the academic front that my opportunities for clinical research, things like that, were much more in the sarcoma realm because as you know, they're rare diseases for which, we don't have great answers. And so we ended up having, I ended up just finding opportunities which worked for me, and the rest is history.
Oh gosh, so there's just a lot there. So that's quite interesting because you and I probably came in an era where we all came as general oncologists, did one or two, three, four tumor types, and we all kind of just stumbled on just doing sarcoma for various reasons. Just like you said, when your decision not to do pediatrics because of different components of family, parents, etc., breast is great. There's a lot of field there, but as you know, the vast majority are generally women, although men, as you said, can get it. Um, and as you said, the space is quite open for sarcoma. Well, it's quite crowded in the breast and other areas. So it wasn't but a big grand design that you went into sarcoma, but eventually it just fit you and your style and what you want to do and could do and use your talents that way. So that's how I'm hearing it from you. Is that fair enough, Meg?
Yeah, no, I think it's fair enough. I think, the I just sort of felt like the opportunities I had in terms of clinical research, developing IITs, were much greater for me in sarcoma.
Yep. And to be honest, for those that are listening, it is still the same today. The field's right open. We just love more sarcoma oncologists or people that are interested in this. Meg, you did something very similar to what I did. We trained at the same institution that we now spent our career at. And maybe you can share with our audience here how that decision was made, number one, to stay there, and kind of how that's worked out for you.
Yeah. Um, so I will admit that I initially was planning to leave, partially because of a relationship I was in. And at the end of the day, that relationship didn't work out. So I ended up doing actually an extra 6 months of fellowship so that I could stay and join the faculty, which in the end, I am very happy happened. Um, and then subsequently, professionally, I felt like I've had every opportunity to grow and do things that were interesting for me, that I enjoyed doing. So while I have looked at other opportunities, I haven't I haven't chosen to move. Part of that is because of family considerations. It's not just me. I have my husband and our kids, and so it has made sense for us to stay where we are and not move. And I think my goals in terms of what I do in terms of my career have been just to do things that are interesting. And through that, I've been able to have opportunities and be promoted. I haven't necessarily set out to, gain the next promotion. So it's just worked out, and that's been great. So that's, I haven't necessarily felt like I needed to leave to gain a promotion.
That is correct. As you know, there are many reasons why we all stay in the region. And there are reasons like I've stayed at Mayo for my career as well. And it is not because i- the desire to do, we're both able to do what we want to do in the institution we are at. And that's worked out well for us. For other people, it hasn't worked out. They have different ideas, and we've had some podcasts with individuals that enjoyed where they were, and they said, it's now time to change and do different things. And for you and me, we can do different things even at our own institution. To that point, looking at your CV, I, I was amazed because you've stayed at Fox Chase for so many times. You've been involved in a lot of committees at Fox Chase. And some of us really love meetings, and some of us really don't like committees and meetings here. And maybe you can share how and why you've decided to be an active participant in the various committees at Fox Chase. Because I think maybe you can just share with our group here kind of the committees and how you decided that that was an important thing as service to an organization.
Yeah. So I will say, one of the things about Fox Chase is that it is a smaller organization. So there are fewer of us around to fill those committee roles. So we sometimes do more than maybe others would do at other institutions. So I would say, so I have three different buckets of things that I do. I have my role in the Cancer Center, which is the Associate Director for Clinical Research, and my role for the Office of Clinical Research. At heart, and one of the things that I learned very early on was the importance of clinical research, and so that's been a passion of mine. And so, working with the institution to try and ensure that we do it as well as we can and provide clinical trials for our patients is, I've just always enjoyed doing that. I also have a teaching responsibility with fellows. We have residents who rotate through for electives, but they are not in the hospital here. And so one of the having fellows working with you, we always participate in sort of recruiting of fellows, and so I have that aspect of it. And then I also, in terms of the department, have had taken on roles. I'm actually one of the vice chairs for the department. Those are sort of the three buckets, and with them comes sort of a set of things that you do. Many of them, some of my roles as vice chair sort of overlap with education, with clinical research as well. So, and yeah, so I think about that day to day. And then I guess I'm a clinician as well, so got to take care of those things.
Well, a lot of us are being told over time, focus on your career, which is important. And then because you only have so much time in a day, you have three buckets that you're trying to juggle here. And, you've done a great, successful job. And then, you know, you've sunsetted some of your clinical, some of committees and you moved on to, it's not that you sunset them, you went to a different one. And then it's all these different ones. And I think that is service to the organization that sometimes we all forget. That's important as we develop out our skills as, seeing patients, being clinicians, being teachers, and then, that aspect of service. So we do appreciate that. And I hope Fox Chase appreciates all the effort that that you all put in there.
Yeah, no, I feel appreciated. So, but
Well, that's the other part too, because sometimes you participate in these committees and at various reasons you think like, why am I doing this? My voice doesn't get heard or I don't have an impact. But if you're small enough and you have, you have a purpose, you can be heard and you can make changes. And that's the part I think you've done too over time at Fox Chase. Just like we all had no real plans to do oncology, specific sarcoma, how did you end up being somewhat tied to, of the hundred of histologies that you could have picked, how did just kind of end up in your wheelhouse? And for people that are thinking about it, maybe tell us a story of how that happened.
yeah. So, it started by being able to participate in the Novartis-led phase 2 trial of STI 571, which we now know as Gleevec or imatinib. And I have to thank Bert Eisenberg, who was the chair of surgery at that time, who, having heard, remember Bert Eisen? Yes. Um, he, was reading the literature, saw this discovery of the KIT mutations, heard about STI 571, and, sent in an LOI that I helped, write to Novartis. There were several other investigators that did, and we ultimately ended up with this phase 2 trial. That was such a groundbreaking change in the management of just, it was very much a lucky break that was a great career starter. At the time that happened, sort of in the clinical research realm, Bert and I convinced one of our basic scientists or translational scientists, Andrew Godwin, who is now at University of Kansas, to dip his toe into doing GIST research. And he had several talented postdocs who came through the lab, both of whom were awarded K99s. And we were lucky enough that one of them, Lori Rink, stayed on and is now an associate professor in our uh, uh, cancer center, and her lab is primarily focused on GIST. And so I was one of her mentors on her K99, and we have continued and have an ongoing relationship, and are co-funded in terms of doing research in GIST. So it was just sort of this opportunity to start at a very early time in the development and understanding of GIST biology, and then subsequently both within the institution, and then I would say also multiple other opportunities outside of the institution. The NCI NIH wild type GIST clinic that I participated in and really collected and described much of SDH wild type GIST biology. Worked with a group that was funded by an organization called the David Foundation on PGFR alpha GIST. And from that, some interesting biology and discoveries came about. So just with all those interconnections, with at the very beginning, after it was clear that Gleevec was so effective, the development of the two trials, both in the United States and Canada, S0033, that was led by SWOG, and then the EORTC trial that evaluated 400 versus 800 milligrams of Gleevec. Being going to the NCI and presenting the data, and having a trial put together, largely led by George Dimitri, essentially within, I think the study was written in about 3 weeks. So I was just there. It was a really exciting time to be there. I'm proud to say that I was one of, I think, the only woman at the time in the United States who was part of that team and group. I'm thankfully joined by many others now. And it was a really fun and exciting time, and it's just kept going.
So, what keeps you in the field of GIST?
There's still unanswered questions. So, there, through the study in GIST that I first started in the year 2000, and I left my honeymoon and flew to ASCO, where we had our first investigator meeting before the study started. And so that was the year 2000. And since that time, there have been all the studies that have led to approved drugs, sunitinib, regorafenib, ripretinib, avapritinib, and also studies that haven't led to approved drugs. Many other drugs that I participated in, nilotinib, ponatinib, looking at HSD90 inhibitors, just pulling those off the top of my head. That there has always been clinical research work to do. Then there's always the question about, why do some patients respond and others don't, and understanding resistance. There's a lot that's around different mutations, but there's also probably other biology, and that's a lot of what I work on with Lori. There are different subtypes, SDH deficient GIST, NF1 GIST, that we still don't, for those cases that we see, don't necessarily have the best options for. So still lots of questions to answer. And now that we have all of our effective therapies, we've, as with many TKIs in sarcoma and other fields, we have to deal with resistance. And so how do we overcome resistance, and what's the best way to do that?
Absolutely. So those that are listening and just don't want to give the impression that things just fall into Meg's plate. She's very active in pursuing a lot of activities to make sure she's at the table earlier on. And it's not by accident that she became the expert in GIST over her career. So, Meg, you talk about, GIST, imatinib, Gleevec, STI, as you said, changed the landscape for sarcoma forever and a day. Prior to that time, we were lumped in with melanoma. We had a drug. Yeah, it was one drug for one histology. Yeah, we tried it for every single histology, as you can imagine, but it did set our course separate from melanoma. And I think part of your career path is that you did change the direction. However, because GIST and all your work has been so effective, a lot of community oncologists now are able to manage very appropriately gastrointestinal stromal tumor. As research groups, as academic centers, what advice would you give to us as academic centers seeing patients that are reverted from community? And what advice would you give to the community oncologists? Because there's a lot of nuances around GIST, and just some general thoughts, because it's almost out there, out in the community, that almost anybody in oncologist can manage GIST.
Yeah. So I think in taking care of a GIST patient, the most important thing to do when you first meet them in their first diagnosis is to know what their mutation is, or what's the driver for their particular GIST, because that changes how you take care of them, or can change it. I think that as we move on to other drugs beyond imatinib, which for many patients is the best tolerated and does the most for them in terms of controlling advanced disease, the drugs become more complicated in terms of side effects and how to manage them. Many of these drugs are used for other diseases as well, so it's not as if it's unique to GIST. I think one of the secret sauces that I think many GIST oncologists or sarcoma oncologists who see these patients is that, we work really hard to try and maintain patients on therapy and manage their side effects. And there are lots of patients that I see who, are on the drug for a month and then they can't tolerate it. And so what does that really mean? Because we do have other drugs, but we have limited other drugs. And so are we throwing away potentially an effective drug that might provide this patient benefit? So I think in terms of thinking about how should community oncologists think about their GIST patients and taking care of them, first of all, make sure you check the mutation. And then, if you're having challenges with maintaining or taking care of your patients on lines of therapy, seek out an opinion to say, is this the right drug? Am I doing something? Could I be doing something different that would help the patient? I think the other thing is that there are increasingly interesting trials that are looking at newer drugs that are being moved up to second-line therapy. And so having a sense of what are the trials out there and does this make sense for my patient? Are they potentially interested in having this opportunity to receive a medication that they may not be able to receive for many, for 2-3 years down the road because of the process of the clinical research. And I think that's where clinical trials are an opportunity for patients. And many patients um, hear about them, but some don't. And I think part of the oncologist's role is to make sure that we're evaluating all the potential therapeutic options a patient has.
Yeah. Well, that's sage advice, so that's very, very important. Yeah, I think mutation analysis is what you're saying, recommending, and managing the side effects. And the key part is, we do have algorithms, pathways that says boom, 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 boom. But what you just said too, it's important to realize that we haven't solved it. There are still issues and a lot of good information that we don't know yet, and we still are doing clinical trials in this space. To that point, early in your career, you were very engaged in cooperative group activities. You've done work at Fox Chase, but also there's organizations on your CV that I even forgot that existed here, and it's good to hear. So maybe you can kind of share with our group here why we needed ICAS, and you can share kind of what ICAS was, and then back in the day, we had a sarcoma ECOG working group, which you were the chair, and those things have evolved over time. And maybe you can hear some history of why all that was needed and your thoughts on where we are.
Yeah, so I think the GIST study demonstrated the ability of doing cooperative group trials together in sarcoma. And so following that, I think there were efforts in the different cooperative groups at the time. It was SWOG, it was ECOG, I'm trying to think who, the surgical ACOSOG group, and work to see how could we better work together and try and do studies. And so ICAS was sort of this approach to try and have sort of an intergroup, if you will, of sarcoma working groups, make it sort of I would say that there were some great studies that were developed and that tested some agents. There was a study looking at EGFR inhibitors in MPNST because of a signal there. I did a study that looked at sorafenib in a variety of histologies. And so one of the challenges, though, was this was before the days in which we had the central IRB really looking at studies, all types of studies. I think they had just started. They were only looking at phase 3 studies, and most of the studies we did were phase 2. And so essentially what ended up happening is whoever did the study in whichever group it was from, it sort of ended up being that group's study. And it really didn't meet the objective of being an intergroup. And ultimately, it lost its funding. And I think some of that was actually sort of taken up by SARC, which sort of developed in around that time and sort of took over this idea of trying to do collaborative clinical trials with multiple institutions.
Yeah, you're right. So there was a void. We tried to fill the void. I think SARC has filled a huge void at that time. There's a big effort now to have sarcoma working groups within the established CTEP groups or committees as well. And it's an exciting time that people are still interested in building careers in this space. So it's a fun time. But yeah, back in the day, we didn't have it, and ICAS was one of them. And I would say for the group, it's called the Intergroup Coalition Against Sarcoma. So that's called ICAS. So good. To that point, there's a lot of pathways that you've developed over time. And one of the pathways people do look at for guidance of how to do things is the NCCN guidelines. And maybe you can share what your role is in that space. And then more importantly, how did you get involved with that? And where do you see that going?
Yeah. So NCCN was developed I, by comprehensive cancer centers, really with the idea that, you know, they wanted to provide the information for insurance companies about decisions about care. And so developing pathways about how we should be taking care of patients was sort of the model. So there were, I don't remember how many initial member institutions they were, but there were member institutions. Fox Chase happened to be one of the founding members. And so, for each of the committees that were developed, a member of the institution is asked to participate. And so I was asked to be the member from Fox Chase for the soft tissue group. And so I was on the committee for a period of time. George Dimitri was the initial chair for the committee. And when he stepped down, he, I think, suggested to the committee, to NCCN, that I take on the responsibility, and I have done so since then. It's a lot of fun in the sense that you get to interact with people from various institutions who sometimes have a slightly different approach to how you do things, and I'm always learning things from my colleagues from at the meetings, and trying to develop guidelines that are evidence-based. Sometimes it's challenging in sarcoma because of the nature of the rare disease and the fact that prospective large clinical trials, randomized phase 3 trials, are not done in all disease settings.
So you kind of, first of all, thanks for your leadership in this space and trying to corral the whole sarcoma comprehensive cancer centers to develop guidelines. But you did suggest that the main purpose at one point was to provide insurance companies enough data to say that this should be an approved drug for your company or your insurance plan. And you always try to balance getting approval for insurance because that's ultimately for our patients, but you're also balancing the data as well. And I know there's a big push to be more proscriptive of the guidelines, but you got to know what it's intended for, and how do you balance that?
Yeah, I think part of it is looking at the data, and there certainly are, and I think for me the biggest example is the role of immunotherapy. I think there are certain centers and portions of this country where immunotherapy is a very attractive treatment. And the data that we have some data, but it's limited data. And so trying to justify appropriately the use of immunotherapy in certain contexts, versus saying, every patient who has sarcoma should at some point try immunotherapy. I don't think that's appropriate care. And so I don't think anybody does, but trying to make sure we have an appropriate balance. And so I think that, takes a discussion. And there is also sort of this, the guidelines have certain levels of recommendation based on the data that's available. So we do also, look at the information, and then we have to vote our conscience in terms of making recommendations, especially when it's controversial. I think the process works. I think it's interesting that every year when you look at the guidelines, you find things that are, you missed the year before, or there's new data that you have to incorporate. So it's always changing. And as it's getting more complicated, how do you share that in a way that's meaningful and measurable, that the audience that's reading the guidelines can actually understand it. So.
Well, it takes a good amount of time and effort. We appreciate it, and also your your basic character of how you approach life does help out the committee. So we do appreciate that. You know, I should be not remiss, but I think you were a PI of a SARC study. Is that correct? Many years ago, and um, that was a long, and I think the number was 22, and we're higher now. We're about into the 40s and 50s, maybe. So maybe you can share with our group here how you got interested in that, engaging with SARC at that time. And what would you tell young investigators or colleagues that are thinking about how to navigate all the opportunities? Because you've lived through all the opportunities, and how would you direct somebody?
Yeah. So, I mean, writing an IIT is fun because it's your idea and you get to explore it. The study looked at a drug called lincitinib, which targets IGF-1R, and our lab at Fox Chase had identified that there was clear overexpression of IGF-1R in um, what was called then wild type GIST, and it appeared that at least in the models we could test, which were not SDH deficient lines, that there may be some benefit of that type of targeting. And so thus sort of came the idea of trying to develop a study using an agent that would target IGF-1R. I had applied for an R21 grant from the NCI, and I collaborated with the group at Dana-Farber for using some interesting PET imaging to sort of see if we could understand this drug that potentially could affect the insulin pathway. How does that affect imaging with PET? So that was sort of the focus of the grant. I initially applied with one agent, which was an antibody that targeted, and the sponsor of that agent withdrew it and didn't continue to develop it. So I was then fortunate enough to be able to get it through CTEP with lincitinib, and almost didn't get it because the maker of that drug decided ultimately with the CRADA that they were only going to support 2 studies through CTEP. And I think because of potentially R21, that helped it. And working with SARC, we ended up doing it that way. There had been another proposal by another investigator in that space, so it sort of took over that. So it was it took a lot of work. I think anybody who's worked and developed an IIT, the story of getting your drug and making sure that you can have it for your study is not a new one, and I think that's one of the biggest challenges. And then then there's the challenge of funding the study. Certainly, the R21 provided a lot of the support for the correlative studies, but SARC did contribute in terms of the funding of the sort of the clinical research part of it, and so it was recruited well. We met our goals in terms of patients. It was a stage 2 design. We didn't pass stage 1, unfortunately, but here we are, and it got published, and yeah, it was a great collaboration.
Yeah. You bring up the point there that you owned it. You had the IT, you had to work through it, you had to get the drug from CTEP. Yeah, you had another monoclonal. In that era, people were thinking insulin growth factor receptor was a key to a lot of things, and you pursued it. And I think what I'm hearing from you is that there's just not one pathway to do studies. You talked about your relationship with George Dimitri and that team at Dana-Farber. You've done a lot of collaborative studies in that space. You've led inter big, intergroup studies and in there, and you did the small earth studies as well. So as you are advising young staff or junior staff, do you advise them to just find out what's available and just try to plug it into a different area, or you tell them no, from our grant we need to do CTEP trials, or how do you coach people?
Yeah. So I use the line that my mentor, when I was doing immunotherapy, used to say, and Lou Weiner, who's now the cancer center director at Lombardi in Washington, D.C., he would say, at the end of the day, you want to, work on, and so that if somebody hears the name Meg von Mehren, they will say, well, she's the gal who, or he's the, if it's Scott Akuno, he's the guy who. So I think when you're starting out, you don't know if your first project is going to be a winner, so you can't necessarily put all your eggs in one basket. But I think you do sort of want to curtail your efforts and not do 5 different things in 5 different areas, and try and sort of have it in some way integrated. I think it's really helpful if your research is integrated with your clinical care, because in clinical care, you see things and it stimulates conversations or ideas that you can maybe then ask questions that can be tested in clinical research. I think the other thing that I didn't appreciate as much as I do now, um, when you're starting out, you look to your mentors. They've achieved. They're doing what they're doing in their A game. Uh, and I felt like I had to be like that person and doing it on my own. And so one of the things I always tell people is it's not a sin to collaborate. You actually will go farther because you'll probably be able to do bigger things and ask bigger questions because you're collaborating with more institutions. You can do a bigger study because you'll be able to accrue more patients. And so the importance of collaboration and mentorship can happen. The other thing I say, mentorship can happen locally, but there are also mentors out there at other institutions that developing collaboration with them and getting their input is also something that one should do if that's where your research leads you.
Great advice. As I'm just stopping to reflect and preparing for the talking to you, even though I've known you for, since our careers started together, looking at what you've done, your CV, and if a young person comes there, they're going to think, "Oh my gosh, how am I going to do that?" Well, it took 30 years or 20 plus years for you to get to where you are with those collaborations. So, hey, find your niche, kind of work through it, be collaborative, and work your way through. So, I think that's great advice. It's good to know. Like, I'm not saying that we're done with our careers, but we have to also start to think about what are we going to be looking at Fox Chase or in the sarcoma community with Dr. Meg von Mehren in the next 5 to 10 years? What should we be looking for?
It's a great question. So my husband's semi-retired, so I sort of see what that life is like. And you know, there are many things that I'm still excited about doing, but then I also think, it would be nice to be able to be more relaxed at times and, not have to do epic notes all the time. so, um, uh, so, uh, so I keep thinking about what is it that how will I, pass on some of my responsibilities, thinking about, succession planning for the many things that I do. And so I think that's part of it. Doing, for me will be stepping down from some of my responsibilities and handing it off, hopefully, to um, uh, people who will take it on with their younger energy and new thoughts and ideas. Uh, I also think about a lot of the research that I'm involved with, which is still exciting. Some of it is in the lab, some of it is, in clinical research. And so, how much more time do I want to spend doing that? And right now, I don't know what that answer is. Still uh, putting in or thinking about new concepts and new grant proposals and things like that. So I'm not quite there, but at some point, it would be nice to be able to have more leisure and um, do things. There are also certain things that are important to me that I would like to be able to spend maybe more time advocating for that, I don't necessarily now because of my work responsibilities and the things that I do. So there are other things that I could do beyond um, medical oncology.
Oh, there you go. So all of us are thinking of glide paths along the way, but what I'm hearing is no, you're still in the fight. You're still putting in grants and that, and we'll see what the next 5 to 10 years means for Dr. Von Mehren and you. And I mean, you are Dr. Von Mehren, but from our sense that yeah, we'll see. And it's important to kind of know that there will be a time when younger staff, junior staff, senior staff have to take over new responsibilities, and they'll come up with new perspectives that will hopefully push us further along. Um, Actually, not only further, but farther down the path. So this is great. So, Meg, thanks for spending time with us today and sharing your time with us. Um, you provided countless amount of time and effort for your patients. I know that. Personally, I know mutual patients have come back and forth, and you've supported many junior staff that have gone on to careers elsewhere. Many of us know those. But do you have any final comments for us as we close?
You know, as medicine gets more challenging in some ways, sort of the infrastructure for medicine and what we have to do, I think it's important for us all to sort of take stock into why we're in medicine and what it is about taking care of oncology patients that is important for us to get up and do every morning. I think sometimes the stresses or pressures that are there sometimes make us think about, should we keep going in this field? And I think there's lots of reasons to keep going, and certainly lots of interesting questions to still keep answering. So I'm hopeful that there continue to be people who want to come into the field and find the next breakthrough for one of our many tumors that we are still struggling to take care of.
What a great way to end. The struggle is still worth it. There are many unanswered questions, and we hope that you continue with that struggle for years to come. So thanks, Meg, for spending time with us. So thank you.
Thanks, Scott. It's been a pleasure.
Perfect. So for those that are listening, this will be coming out sometime in February of 2025. We look forward to further conversations we have at SARC Talk. So thank you all.
Thank you for listening. Be sure to subscribe so you never miss an episode of SARC Talk. To find out more about SARC, please visit our website at sarctrials.org. To suggest a topic or for questions, please email us at sarctalk@sarctrials.org. Together, we can find a cure for all forms of sarcoma.